Without further ado, we'll introduce our moderator for today, Dr. Emily Cole. Emily is the Associate Director for Research Relations at the Computational School of Engineering at UT Austin. Take it away, Emily.
Thank you, Javier. Hello, and welcome to our newest webinar in our Text Talk series. Today's event will focus on next-generation imaging in science and medicine. If you are new to our seminar series, the format will first include a presentation by our speaker, followed by a moderated Q&A session where we welcome questions from you, the audience. We ask our audience members to please type your questions in the Q&A box throughout the event, and we will answer them immediately after the presentation. It is my pleasure to welcome today's speaker, Dr. Sheto Chaudhary. Dr. Chaudhary is an assistant professor in the Department of Electrical and Computer Engineering here at the University of Texas at Austin. His research interests are in developing next-generation optical imaging technologies for applications in science and medicine. A key emphasis in his work is the joint design of novel optical imaging systems and advanced computational frameworks. This co-design of hardware and software enables imaging capabilities not possible in traditional optical imaging systems. Dr. Chaudhary's work on deep tissue imaging is currently funded by the Chan Zuckerberg Initiative, and he was recently named a 2021 Fellow for the New Skylog Initiative, Advancing Bioimaging. Previously, Dr. Chaudhary was an NIH postdoctoral fellow at the University of California, Berkeley, in the Department of Electrical Engineering and Computer Sciences. Before that, he received his PhD and Bachelor's of Science degrees from the Department of Biomedical Engineering at Duke University. I'll now hand it over to Dr. Chaudhary for his talk titled Next Generation Imaging in Science and Medicine.
Okay, thank you so much for such a kind introduction, and thank you all for joining me here today. So let me just share my screen. Yeah, so I'll be talking a little bit about today some of the imaging research projects that I've worked on over the past few years that I think are a subset of some of the more general next generation imaging techniques for science and medicine. And so many of the projects that I'll be talking about here will be dealing with things that I've done during my postdoc as well as my PhD time. And so I definitely want to acknowledge my previous lab as well as my collaborators. So optical imaging platforms, there's a couple of them, and they share the trait that they all operate by using light to form images. It's one of the oldest ways we know how to form images, and it's the way that we humans also visualize and understand the world around us. And so traditionally, these imaging systems improve over time by advancements in their hardware design. So here I'm showing an example in a camera as well as a microscope of how their lenses are increasingly complex over as they get developed. And so these lenses require very careful fabrication, design, assembly, and so on and so forth. So this is traditionally the way these imaging systems improve. But over the past few years, really leveraging the advancements of very accessible but powerful computing powers, a new paradigm in imaging has come up, which really rely on the joint design of physical hardware alongside computational reconstruction. And the idea is, by jointly designing these two sides, you can get imaging capabilities that are otherwise not possible. And this new strategy has really affected all types of imaging. So photography, astronomy, medical imaging are all benefiting from this. So my specific specialty is microscopy. And so the goal that I've been pursuing over the past few years is how can I develop computational microscopy systems to target some of the open challenges in science and medicine. Whenever you design an imaging system, you often have to be extremely cognizant of what the imaging needs and priorities are for your experiment. So do you want higher imaging resolution, high speed, depth, multimodal compatibility, low cost, and so on and so forth. And so oftentimes the challenge is figuring out how to design an imaging system that navigates this domain in a way that's optimum for the experiment that you care about. And so I've listed here 3 of the challenges that I've tried to tackle over my career that deal with me trying to find computational microscopy solution. So I'll go through them one by one. So the first one is multi-dimensional super resolution imaging. So traditional imaging systems, optical imaging systems, are resolution limited by diffraction. And so one of the questions that I ask myself is, how can I design, or how can we design a computational microscopy system that surpasses the diffraction limit across multiple dimensions of measurement? The next topic is affordable high-throughput imaging. So most imaging techniques that can image at very high resolutions across very large fields of view require very expensive imaging lenses, as well as scanning stages, and so on and so forth. And so I was wondering, is there a cheaper alternative where you use computation to really leverage computation to get really large fields of view with high resolution in a very cost-effective manner? And the third topic that I'll talk about is imaging through optical scatter. So biological tissue is extremely scattering, and so light cannot travel more than a millimeter or so before it scatters too much to be able to be imaged well. And so this is a problem because a lot of biology is large, and I was wondering if there was a way we could build a computational microscope that is more resistant to these types of scattering effects. So I'll go through these one by one. But before I start, I do want to mention that the first two topics deal with multi-dimensional measurements in the form of different contrast mechanisms. And so one of the biggest benefits of optical imaging is that you have a host of different contrast mechanisms to choose from that gives very informative information about the sample. So you have fluorescence, spectroscopy, Raman scattering, so on and so forth. So the three contrast mechanisms that I deal with are fluorescence, phase, and refractive index, and they each give very important dimensions of measurement, which I'll talk about right now. So just as a quick review, in fluorescence imaging, you have a sample that's tagged with fluorophore molecules, which have the property that when they absorb some excitation energy, they emit at a different color. And so if you detect just this emission, you get this really beautiful molecular specific and background free image of the positions of the fluorophore molecules in your sample. Phase imaging is different. So in phase imaging, you illuminate the sample with a collimated beam, so with a flat wavefront. And because of the internal refractive index distribution of the sample, these wavefronts deform. And so if this is a transparent sample, like most cells are, you see almost no contrast. But if you can detect these phase wavefronts coming from the sample, you can get a really nice high clarity image of the cell morphology. In this case, I'm showing it in color just for visual clarity. But one of the beauties of this technique is that you do not have to label the sample, so it's less invasive than fluorescence. The light can be very weak, so you have little to no phototoxicity for very high signal, and you get quantitative biophysical and biochemical information. And the key point to note here is that both the object here in the fluorescence image and the phase image are the same, but they look completely different because they're using different dimensions of measurement here, phase and fluorescence. And so this is just to illustrate the fact that if you can measure objects with different dimensions of measurement, then you have a much greater ability to understand what's happening in the object and a much greater ability to understand the science of what's going on. So I'll be talking about these challenges and just a little quick sneak peek about my solutions, which I'll go over right now. So for multidimensional super resolution, some of the things that we introduced here was a system based on structured illumination that enabled us to get 3D fluorescence and refractive index tomography. And so we'll talk about that. In affordable high-throughput microscopes, we actually built a system that was so affordable that it used a Scotch tape to essentially allow us to computationally increase the resolution across a whole field of view to get gigapixel scale images. So that was a very promising solution for high-throughput microscopy. And for imaging through optical scattering, we will show some really interesting preliminary results that show promise in reconstructing 3D refractive index tomograms of multiple scattering samples. So we'll talk about each of these one by one. And so I'll start here with multidimensional super resolution. So So before I start that, let me first introduce where the resolution comes from, where the resolution limit comes from in traditional microscopy. So usually for optical imaging, points on the sample map to points on the camera based on a system of lenses, which is considered to be your optical system. And this system of lenses comes with a limiting aperture, a pupil function, that controls whether or not you get a high resolution image or a low resolution image. And so the larger this pupil function is, the higher resolution you get, and the smaller it is, the lower resolution you get. And the cutting edge, some of the highest resolution imaging lenses in microscopy, they give you resolutions of around 250 nanometers. And so this sounds like a lot, but it's actually not enough to see some of the really fine and really small biological components that are extremely important for many phenomena and biological dynamics. So in response to this, a whole host of super resolution techniques have come out that specifically target being able to beat that 200 to 250 nanometer resolution limit. And so they're typically divided between fluorescence imaging and phase imaging. And one of the issues that we noticed a while ago is that they're mostly unidimensional. So they usually operate on fluorescent imaging or phase imaging, but not both. And so the question that I was considering, that we were considering a while ago, is, was there a super resolution technique that could be compatible with multiple modalities, with specifically fluorescence and phase? The answer to that is actually yes. And one of these techniques in this list, structured illumination microscopy, is compatible with multiple dimensions of measurement. And it actually happens to be a computational technique. And so it's a very cool technique. I'll just quickly describe it in an intuitive sense on how it works. So if you have an object pattern, here I'm showing an object pattern here, and if you illuminate it with a structured pattern like this, and let's say the object pattern is so high resolution that you cannot directly see it with your camera, but the illumination pattern, you know what you're illuminating with. The intuition is that if you overlay them on top of each other, you see these really low frequency beating patterns, which are called the moire fringes. So these are observable by your microscope. So if you measure what these are, and if you know what you're illuminating with, you can actually computationally reconstruct your object pattern, even if you can't directly see it. So that's a very simple, intuitive explanation of how this works, but it's extremely powerful because it works for any dimension of measurement you choose. And so we built a system a while ago that essentially illuminates a sample with light. And so the sample is a fluorescent sample. So part of the light just passes through and diffracts through. And part of the light gets absorbed and re-emitted as fluorescence, which is also passed through and then detected on a different camera. But the key component in this microscope is that there's a spatial light modulator that is essentially that's essentially projecting a stripe pattern onto the sample. And so you get raw acquisitions that look like this. So if you look at any one of these acquisitions, you see that it looks like a sample with stripe artifacts on it. But if you look at the whole data set, those stripes are actually encoding this super resolution information into your measurements. And it allows you to reconstruct the super resolved information post-acquisition. To give an example of how it may look, here I'm showing a phase image and a fluorescent image captured with conventional means. And so after the phase image is processed with this type of computational approach, you go from an image that looks like this to an image that looks like this. Oops, let me do that again. From image that looks like this to this. And the fluorescence image, this goes from this to this. And so one of the key, so the fluorescence super resolution has been around for a while, but one of the key contributions or novelties that we were excited about is that this structural illumination actually allows you to increase the resolution in both phase and fluorescence. So it's a multimodal technique. And it doesn't only work in 2D, it also works in 3D. And I'll talk about this right now. So phase imaging, phase is a accumulation of the refractive index. So it's essentially a projection through the sample. So one of the difficulties in phase imaging is that you don't get much 3D resolution using the traditional methods of phase imaging. Yeah. Now, this is actually very similar to computed tomography, where each X-ray in the computed tomography is a projection through the sample. So similar to that, if you can view the sample through different angles, then you can actually have some ability to reconstruct the 3D distribution. Now, so this actually has already been done before, but one of the novelties here is that if you combine multiple angles at the same time and they're mutually coherent illumination, then that is equivalent to illuminating the sample with a stripe pattern, because the stripe pattern is an interference pattern between multiple angles. And so by illuminating with a structured pattern, you can actually also achieve these types of optically sectioned 3D volumes. So that was pretty exciting when we first saw this happen. And we implemented this on actual cells. So these are adenocarcinoma cells. On the left-hand side, we're showing a Z-section fly-through of a reconstruction based on 1,000 sinusoidal illuminations. And on the right-hand side, we're showing the phase reconstruction from just a single illumination beam. And so we see that as we go up and down in Z, we get very little optical sectioning with just a normal phase image, while we get very nice, much better optical sectioning with the sinusoidally illuminated image. And so this has a lot of applications for when you try to identify biological components in 3D, and you see that this type of optical sectioning gives you a much better ability to identify how the refractive index of different structures in the cell are positioned relative to each other. And the greatest ability of this is the fact that this is compatible with both fluorescent and refractive index dimensions of measurement. So you can gather all of these really large data sets based on different dimensions of measurement that allow you to do all of these powerful cross-correlative studies on how the molecule dynamics in the cell might interact with the biochemical or the biophysical dynamics of the cell. So that's an extremely powerful ability that's gained by doing these types of powerful multimodal studies. So that's kind of the spiel for the first project I'll talk about, the multi-contrast super resolution. I'll go on to talk about the next one, which is affordable high-throughput microscopy. So let me first briefly introduce the idea of high-throughput microscopy, where the objective is not so much as it is to get to see the smallest possible thing, as it is to see as much things as you can. And this is extremely important in studies where you may want to see how different biological components that are very far spaced from each other may interact. And so on the left-hand side, here's a beautiful example of a fluorescent high-throughput microscope that's being used to visualize the neuronal structure at different locations within the brain. And on the right-hand side, this is an example of phase high-throughput microscopy, where you can see how different cells in different locations across a wide field of view are undergoing their dynamics. One of the things you notice is that the fluorescent high-throughput microscope is actually quite complex. It requires very careful engineering design, pretty expensive stages and microscopes, objectives. And in contrast, the phase imaging system is typically just an LED array on top of a conventional microscope. And so one of the tasks I asked myself is, could we design a computational microscope that combines fluorescent and phase imaging for high throughput visualization? So this is again getting towards the multidimensional ability. And so this is a project that I worked on with Lee Howe over here. And it actually uses a very interesting intuition as well. So if you look at a very high-end, high-resolution objective, they typically come with very small fields of view. So the conventional way to aggregate together a very large field of view, high-resolution image is to essentially scan the sample through the imaging field of this high-resolution objective. So this requires an expensive stage, requires it, it's a slow process, and so there's some limitations with this method. So the solution that we were thinking of is, what about if we do the inverse? Let's say we start off with a very cheap lens that has a very low resolution but a very large field of view, and so we get an image that looks like this. So it's very large field of view with very low resolution, but if we can somehow illuminate some sort of high resolution structured pattern onto the sample, similar to what we talked about before, we should be able to computationally enhance the resolution to something that is equivalent to what we would have gotten with the high-end objective. And this is actually real data that we were able to reconstruct, and it was made possible by using Scotch tape and advanced computational solvers. So we'll talk about this, about the system right now. So the system is actually very simple. You illuminate the sample with a beam of light. The sample is fluorescent, so the fluorescence gets imaged onto one camera while the diffracted light gets imaged onto another camera. But the key component here is this diffuser, which is essentially a piece of two-layered Scotch tape that we're sliding into the camera's field of view and then slightly defocusing. And so when we defocus here, we get a very nice high-resolution speckle pattern that actually acts as the encoding element that encodes in all of the high-frequency information about the sample through the low-resolution imaging lens to be measured later. And so our raw acquisition actually looks like this, where we're essentially sliding the Scotch tape around, and this results in the speckle pattern kind of jiggling around. And so this is what we use to do our reconstruction. And so our cameras in this example were around 5 megapixels worth of information. But based on this type of computational enhancement of resolution, we were able to computationally enhance the 5 megapixels worth of information on the camera to around 200 megapixels worth of information. And there's no reason that we shouldn't be able to do this to gigapixel scale images. So here we're zooming in towards within a millimeter scale field of view towards a sample. So these samples are like 1 micron diameter microspheres. So on the left-hand side, we're showing the native resolution of the imaging lens with which you would normally see the sample. And on the right-hand side, we're seeing the computationally enhanced resolution based on this type of structured illumination. And so clearly we can see that our resolution is enhanced dramatically by a factor of around 5, actually, across the whole millimeter scale field of view. So this was actually very exciting when we first saw this work out. And of course, we can do this for biological cells. So this is HT29 adenocarcinoma cells. Here we're showing how we're moving the speckle pattern from the Scotch tape around in at different trajectories. The reconstruction is also multidimensional. We're seeing the phase on one side, the fluorescence on the other side, and we're going up and down in Z. And so we're getting subcellular resolution of imaging these cells across a very large field of view. And so this was very exciting. And again, this was really enabled by the joint design of the software alongside with the hardware. In this case, we use a simple Scotch tape, but maybe in the future we'll use more well-designed diffuser patterns to allow this reconstruction to be done more efficiently, more optimally, and so on and so forth. So that's kind of our solution that we found very promising for affordable high-throughput microscopy. And so now, I'll talk about the last project for this presentation, which is imaging through optical scattering. And so this is a project that I'm very excited about, and I'll give it a little bit of a motivation. So optical imaging devices form images by focusing light. So if your sample is near the surface of a biological tissue, you can assume that light from the sample comes out in straight lines, which means that lenses can focus light into very high clarity, high resolution images. So that's great. But as your sample goes deeper and deeper into tissue, because of the internal refractive index variations within the tissue, the light will actually start scattering, will start changing its trajectory. And because of this, it can no longer be focused easily with lenses. And so what this results in is a lot of out of focus blur, signal loss, and other types of artifacts that make for pretty poor image quality. And so this is a problem because biology occurs at all size scales. So microscopes are great at imaging within the 100 micron depth range. So cells occur in there, which is why microscopes are used very frequently to image cells. But as you start trying to image thicker samples like skin or small organisms, bulk tissues, or organs, then microscopes really start suffering and they may no longer be the tool for the job, at least currently. And so I always thought that this was very unfortunate because light can actually travel centimeters in tissue before being absorbed. And so I looked at this problem more as a unscrambling problem, like if light can be unscrambled, and then maybe there's a way for us to image these thicker samples. So this was the thought process that started this project. And so what we were doing back then is we essentially came up with a model that, or we extended a model that that essentially decomposed the 3D scattering that's occurring within an object as into a layer by layer summation, accumulation of scattering. And so the idea is, if you know the electric field coming into one layer of the object, then if you know the refractive index in that layer, you should know what electric field is coming out of that layer. And so if you do that layer by layer, eventually you'll figure out, have an estimate of what the electric field is coming out of the last slice of the object. So the Ford model is essentially based on this recursive formula. If you know the 3D refractive index of the sample you care about, you have a prediction of what type of measurement you'll get if you illuminate that object. Now, in reality, you want to do the inverse. You know what measurement you get because that's what you're measuring with your system, and you want to compute what refractive index the sample must have to match your measurements. And so this is a inversion problem, and because it's a non-convex inversion problem, you have to take multiple measurements at different angles or different patterns, and so on and so forth to make this problem more well-posed. But if you do things correctly, you get some pretty promising results, which I'll show here. So, so the optical setup that we use for this is actually a very simple angle scanning system, where the illumination beam is incident onto a scanning mirror that essentially scans the sample at different angles. And so on the left-hand side here, I'm showing an example of a weakly scattering sample and a multiple scattering sample. And I'm showing the data set, the raw data set that has been acquired by hitting it at different angles. And on the right-hand side, I'm showing the corresponding Fourier transforms. And the reason we use Fourier transforms in this type of work is because they give you a very nice intuitive indicator of how multiple scattering a sample may be. So for this sample, this C. elegans embryo sample, you can see there's a lot of haze outside of these two circles in Fourier space, which is an indicator that there's multiple scattering going on. So let's look at reconstructions. So, I'll first show a reconstruction of a fibroblast cell, which is a weakly scattering sample. So this is a single measurement taken at a specific angle. In the middle column and the right-hand column, I'm showing the center slice of a 3D volume. And so in the middle column, the 3D volume is reconstructed using the traditional tomographic reconstruction techniques that assume a single scattering. And on the right-hand side, we're showing the reconstruction from the multiple scattering algorithm that I just introduced previously. And we can see here that both reconstructions look pretty similar to each other, which is expected because this is a weakly scattering sample. Now, if we go to thicker samples like C. elegans embryo or C. elegans worm, we can see that the traditional method of reconstruction starts showing a lot of artifacts. So in these reconstructions, you can't really see what's happening internally to the sample. But with the multiple scattering algorithm, we can very clearly see the internal structure of these types of samples. So this is a very promising indicator that we're on the right track with these types of algorithms. We actually implemented this on the whole body of a 1 millimeter length C. elegans worm. Again, this is all refractive index contrast, so this is an unlabeled sample. And we can see a lot of these features, so we're traveling down the esophagus, but we can see a lot of these features with very high contrast refractive index signal. And so we're passing the stomach, we're going through the pharynx, and now we're going to go up and down in Z. And so this is a 3D data set. It's a 2 to 3 gigavoxels worth of data. So this is a very large data set. But this is just as an indicator, we can see like with label-free, with quantitative refractive index contrast, the very high resolution features of these types of samples, which are multiple scattering. We wanted to be a little bit more ambitious, so we wanted to go from multiple scattering with optically transparent samples to optically diffuse samples. So we picked a Drosophila fruit fly embryo as a test indicator to see how well our algorithm works. So these are samples that are extremely important for a lot of developmental biology studies and are very well known to be extremely scattering. So here I'm showing an example of a Drosophila embryo undergoing development. And as you can see in the inside, it's very difficult to see what's going on. It's very blurry and very hazy. And typically seeing inside is an open problem for these types of samples. So when we did our reconstruction, we first wanted to point out that the size differences between a Drosophila embryo is very different compared to a C. elegans embryo or a mouse embryo, which we've reconstructed already. So they are so C. elegans embryos and mouse embryos both fit within a 113 micron field of view, but a Drosophila embryo is around 1 millimeter in length. And so they're much thicker, much more absorptive, and much more highly scattering. So this represents a greater level of difficulty in terms of reconstructing scattering. So let's see how we did. So this is the reconstruction of a fixed Drosophila embryo in 3D. So this is us going up and down in Z. So we do see a little bit of haze, which is expected because this is so highly scattering. So we're working to clear that up. But if you look at the center slice, we can see a lot of these anatomical features that show up with pretty high resolution and high fidelity. And so this was exciting to us because typically features in the center slice of the embryo are very hazy and difficult to see using traditional methods. And to see very high clarity images like we're seeing here, oftentimes the embryo might have to be cut open. And so the fact that we can see these types of internal anatomical features non-invasively is a very good sign for future studies where we may want to study like the dynamics of how these embryos develop over time and how different internal components grow and interact with each other during the developmental cycle. So that was actually really exciting for us as well. And I guess like this is where the project is currently, and we've been trying to think about how this type of imaging strategy can be useful in the future. And so I mentioned this in the past a few slides ago, but I'll mention it again. One of the biggest application spaces that I think could benefit from this type of computational correction of scattering is in deep tissue imaging. And let me explain why. So this is a picture of me going into lab and essentially putting my hand in front of like a very strong, essentially a very strong flashlight. And as you can see, light is actually being able to travel through my hand and is illuminating a lot of the internal structures like the bone and the veins internal in my hand. And so my hand is around 2 to 3 centimeters. So that's 2 to 3 centimeters of tissue that light is traveling through. And so it's not that we're not getting any signal from tissues of this type of thickness, but it's rather that this signal is getting scrambled, which is preventing us from being able to image with high resolution internal to these types of tissue thicknesses. So if you are able to figure out a way to get the refractive index of these types of samples, you know how light propagates through the sample, and that gives you the ability to figure out what the scattering potential of these types of samples might be. So and so that's very valuable, of course, in refractive index tomography, which is its own imaging technique, but it has applications in other imaging techniques as well. So two technologies that I think could really benefit from this include multiphoton microscopy and photoacoustic microscopy. So these are both presented as deep imaging techniques, but they both rely on either rejecting multiple scattered photons, which is what multiphoton does, or using a probing mechanism that is resistant to multiple scattering. So photoacoustic uses sound waves or acoustic waves, which are much more resistant to scattering than light is. So both of these techniques could benefit a lot. if they did include considerations for multiple scattering, and that could dramatically increase their imaging depth even longer depths than what they use right now. Optical simulation is another big application space for this. So if you can image a optically scattering object, theoretically, that might also imply that you can excite into an optically scattering object. So that has many applications. So one of the most popular ones is optogenetics, where you might want to stimulate neurons that might be behind or underneath a layer of scattering brain tissue. So lots of examples, and many of these I would want to, I imagine that we might pursue in the future. And so I guess I really do want to convey that this general field of computational microscopy is extremely interdisciplinary. It relies on formulating physical models on how light interacts with matter. It relies on building these imaging devices that can encode information about your sample into your measurement. And it also relies on being able to formulate these computational inverse solutions. So these are extremely data-intensive software approaches, and so they rely on, supercomputing expertise, cloud computing, machine learning, and so on and so forth. And lastly, it also requires imaging analysis research, where a lot of times the outputs of these reconstructions are terabytes worth of information. So if you want to be able to draw a biological conclusion with some degree of statistical significance, it's much better to be able to write code that can automatically identify features within your sample, cross-correlate different patterns, and so on and so forth, rather than having a grad student go through terabytes worth of data to draw that conclusion by hand. And so these are all subfields that are encompassed within the general field of computational microscopy. And I'm basically interested in pursuing anything like this for scientific translation towards scientific discovery. And so, I guess in conclusion, I hope I've conveyed to you that there's a lot of powerful capabilities that you can get if you jointly design imaging hardware with computational reconstruction frameworks. And this allows you to get imaging capabilities that are otherwise not possible with traditional imaging systems. And so our focus is to figure out how we can design and engineer these types of technologies to find utility in scientific or clinical workflow. And so that's kind of the spiel for my talk. And with that, I'd like to thank you all for your attention and take any questions. Thanks.
Thank you, Sheta, for a great talk. So we have a lot of questions coming into the audience, so let's just get into it. First question is, how can you tell if a sample is multiple scattering or not?
Yeah, so one of the ways is using those Fourier circles, where if you see a lot of signal outside of those two regions in Fourier space, you have a sense that it's multiple scattering. A more intuitive way is to see if you see a lot of out of focus haze. If you're imaging the sample and you're trying to image in depth and you see that there's a lot of features that seem out of focus to you, then that probably implies that there's a lot of multiple scattering going on. So it's actually very easy to see in practice. If you can't form a very highly focused image and highly focused and optically sectioned image using normal wide field microscopes, then that typically means that there's a lot of multiple scattering going on.
Thank you. We have one question. I think this is referring to the third project that you were talking about. What is the use of fast Fourier transform?
Yeah, let me see if I can share my screen for that. So I kind of gave a general overview on that. Let me see if I can share my screen. So intuitively speaking, the Fourier transform here gives an indicator of how multiple scattering a sample may be. So I'm showing two examples here of a weakly scattering cell and a multiple scattering embryo. And you can show using physics that if it's a weakly scattering sample, and you're illuminating at different angles and you're detecting intensity, that you essentially see two circles that are rotating around each other. So you can show this using physics. So there's a physical explanation for that. But the physics that describes why there's two circles is broken when there's multiple scattering going on. So if you have multiple scattering going on, you start seeing a lot of haze outside of the two circles. And that gives a very, very intuitive and very immediate indicator of how much multiple scattering there might be. And so C. elegans embryos are kind of scattering and kind of not. But if you go to even more highly scattering samples, you'll see that in some situations you won't even see these two circles. You'll just see a big, circle of big splotch of information. And in those situations, it's very obvious that it's a very highly multiple scattering sample, and that's given to you by the Fourier transform. Yeah. So.
Okay, thank you. We have another question. This is one that probably many of the audience have this question is, how long does it take to computationally reconstruct the images?
Yeah, so that's a very important question. So the C. elegans data set that I showed, which is around 2 to 3 gigavoxels of data, it took us around a week or week and a half to reconstruct. So it's definitely not a fast process. Now, that said, this was done using code that we wrote up in MATLAB or Python. So there's a lot of, and it was a very general optimization-based code. That does not mean that this could not be dramatically sped up in the future by using different types of regularizations, different types of prior knowledge, also different types of cloud computing and machine learning tools. So we're hoping that in the future, to make this very feasible for biologists to be excited about it, we're hoping to speed this up to a time frame that is attractive to these types of end users. So right now, it takes a while, a week or two, to reconstruct 2 to 3 gigavoxels of data. But there's no reason that we can't dramatically speed this up in the future when we get the pipeline down working properly.
Sounds good. Have you experimented at all with planoptic lenses or micro lens arrays?
So we have not experimented with that. I'm interested in experimenting with that. I have a few ideas. We have a few ideas on how we can use micro lens arrays to generate different types of structured patterns. But in short, to answer your question, we have not yet currently used them in the types of research projects that I've talked about today.
Okay, so possible for the future.
Yeah, definitely.
And this is going back to the fly embryo example. How are the light doses and imaging times for that example? Is time lapse possible?
So time-lapse imaging is definitely possible. So I talked about 1 or 2 week reconstruction time. Reconstruction time is different from acquisition time. So although it takes a long time to reconstruct, it's actually quite quick in acquiring the data. So you can reconstruct offline. So to answer the question about acquisition time, it takes around maybe 6 in the images in the reconstruction data set that I showed. It took around 6 seconds to acquire around 300 acquisitions at different angles to reconstruct the image. So at that type of imaging speed, from what I understand, imaging the developmental cycle of a Drosophila embryo should not be an issue.
And this is kind of a question going back to, again, the speed of imaging. So is it possible to apply the algorithms in real time?
I never want to say if something is impossible. There's a lot of, so it might be possible, but there would have to be a lot of development in the computation to try to figure out how to speed these up. As of right now, I am not sure how to do that, but maybe that just means I need to work with someone who does.
Potential collaboration there.
Right.
And then a question, kind of a more detailed question here on the light source for the devices that you're using. So for example, the wavelength, and do you get different results if you alter those properties like the wavelength? We didn't really touch a lot on that. So any details on how you're actually setting this up and what is the source?
Yeah, so that's a great question. The types of images, so the light matter interaction is extremely dependent on wavelength, especially with scattering. So in the picture of the hand that I showed, which is being illuminated by white light, is being illuminated by white light, but you only see red light coming out, which just means that the green light and the blue lights are being absorbed while red light is passing through. So that's a very quick example of how wavelength does matter. Now, the question of how I choose wavelength is also very interesting. So for the C. elegans and the C. elegans embryos, as well as the C. elegans worm, these are pretty transparent samples, and so they are actually transmissive to most visible light. And so the data that I showed for those reconstructions actually utilized a green wavelength, because green wavelengths can get through, and they give high resolution reconstructions. There's a trade-off. The longer your wavelength is, the lower your reconstruction gets. So for the C. elegans, I picked a wavelength that was able to penetrate as well as give good resolution reconstructions. So that was not true for the Drosophila embryo. So I didn't actually talk about this during the presentation, but the green wavelength was too highly absorbed with the Drosophila. So with the Drosophila embryo, I actually used near infrared wavelength, so 980 nanometers light. So that allowed me to get enough signal through the embryo to do the reconstruction. And and another benefit was not only was the absorption less, it was also less scattering. So it made the iterative reconstruction a little bit more easy to solve and a little bit better posed. So yeah, so wavelength does matter. As of right now, I basically picked ones that happen to work. And I'm sure that in the future, we'll probably have more rigorous ways of figuring out what the optimum wavelength is.
And this, I wanted to jump to this question since it's pretty related to what we're just talking about. So for the deep tissue imaging work, what kind of light source would you prefer there?
So for the deep tissue imaging work in the future, we probably would want to use infrared, near infrared light sources, the type of which is used in 2-photon or 3-photon microscopes. So so right now we're thinking of purchasing a source that allows us to excite with 1300 nanometer wavelength, because that is a wavelength that we saw gives us good penetration into these types of scattering tissue.
And then we talked a little bit about this when we were talking about the speed to reconstruct those images. So what do you think is the future of applying AI imaging in the life sciences? Is this the next big application of AI to kind of help?
Well, I definitely think there's a lot of room to use AI in the life sciences. Well, I mean, there's room to use AI in almost every branch of science. I wouldn't say that life sciences is unique in that role. I would say though that there is one caution that I have with applying AI to the life sciences, which is the existence of, or trying to use AI to visualize things that have not been seen before. So one of the biggest benefits of these new microscope techniques that are not AI based is that they try to allow you to see phenomena that are otherwise not possible. The problem is, if you try to use AI with it, if you don't know what something should look like, it's very hard to know whether your AI system is hallucinating that object or not. So there's many examples of how AI systems have reconstructed objects that are not realistic, that may look realistic but are not realistic. So I think there's a lot of room to use AI in the life sciences, but I also think that there needs to be a lot of validation studies to make sure that the reconstructions that are outputted by AI, even if they look realistic, are actually being validated against some sort of mechanism. So I would throw that out there. So I think there's a lot of room, but there's also a lot of danger as well. So, and it makes sense to be very careful in how you use AI for these types of applications.
Yeah, it sounds like being careful and maybe a real good collaboration between the experimentalists and the folks working on the AI side.
That's right.
Well, it looks like that is about all the time we have for questions today. So I would like to thank Dr. Chaudhary again and our audience for an engaging discussion. As mentioned in the chat box, a link will be sent for the live event in the next day or a few days. If you would like to connect with Dr. Chaudhary, please do not hesitate to reach out directly or to our research relations team. I even saw in the chat box, I think you've already got some undergrads who are interested in how could they join your lab. So we can follow up there. I'll now pass it over to Canavius Smith for wrapping us up today.
Thank you so much, Emily, and thank you, everyone. My name is Canavius Smith. I want to formally thank each of you for joining today's webinar, and we hope the conversation was valuable. If you would like to learn more about future webinars or to hear more about hot topics that are changing the industry, please be sure to follow us on our social media channels. As you have heard today, lifelong learning and technical adaptability is fundamental in preparing individuals to remain competitive in today's environment. If you enjoyed our fantastic speaker and would like to learn more about partnering with the university on research opportunities, please email the Research Relations Office to initiate a discovery discussion. The Research Relations team partners with companies from around the world to solve some of the world's biggest problems in energy, water, transportation, imaging, and many more. The Office of Research Relations works to facilitate and derive high value from research collaborations between your companies and UT's researchers, and they would love to work with you today. Along with the Research Relations team, we also want to thank the two other units of the Cockrell School of Engineering that helped put this wonderful program together. First is the Texas Engineering Executive Education, who works to reskill and upskill companies' current workforce, either through our master's degree programs, graduate certificates, or short courses. So if you need any training around STEM education, please be sure to contact this department to learn more. And next is the Petroleum Extension of Texas, or PTEX. This department has been around for over 75 years, serving the oil and gas industries and professionals with skills training either through e-learning, publications, or online or in-person training. We have the training to meet your energy needs. All three units are here to serve you and your company through education, training, and research. So feel free to contact us at any time, and we would love to collaborate on helping you reach your company's full potential. If you would like to provide feedback on today's webinar or to learn more about receiving CEUs from today's presentation, make sure you complete the evaluation survey in the chat or in your email following this presentation. Once again, we'd like to thank you for joining us today. We hope you have a great weekend and hook 'em horns.